Today we're going to have another one of our specific disease-related lectures on tuberculosis. We're going to have three. So we'll have tuberculosis, HIV, and malaria. And these are the three major infectious diseases. And it's really important to look at each one individually because they're representative of a viral infection, a protozoan infection, and in the case of tuberculosis, it's representative of a bacterial infection. And these three diseases are responsible, probably have the most morbidity and mortality of the infectious diseases that are currently affecting human individuals. So let's go on and discuss malaria. So malaria, again, if you remember in our acid-fast discussion, is an acid-fast bacteria. And it stains. It does not stain with a gram stain. It actually requires a different kind of staining, this acid-fast staining. And the reason for it not registering on the gram stain is because of the cell wall. Now, in a sense, it behaves a lot like the gram-positive bacteria with this external cell wall. But the cell wall is actually much more fortified than even the gram-positive bacteria. And we'll see that this property that the Mycobacterium tuberculosis has enables it to have been around since the beginning of humankind. And basically, so tuberculosis as a disease is spread by airborne droplets. There are two kinds of disease. There's latent disease and active disease. And recalling our discussion about opportunistic infection, that tuberculosis is considered an opportunistic disease in relationship to HIV infected individuals. So in other words, HIV infected individuals are especially susceptible to developing tuberculosis. Now, you can see here kind of a funny picture about this strong man, and this was some of the thinking that was going on in the 40s and 50s in the United States when the incidence and prevalence of tuberculosis was much higher than it is now. So this is a picture of the tuberculosis mycobacterium cell wall in particular, and you can see the extensive width, right, of the cell wall, and these are all lipids, and that's what makes it carry the property of having a very waxy outer coating, and we still have the peptidoglycan just like the other gram-positive bacteria, but we have this extra, it's almost like an extra layer on top of that. So it really makes the tuberculosis bacteria much more durable even than these gram-positive bacteria. So in terms of worldwide incidence, these are the countries that have very high, they're called high burden countries, and these are the countries whose new tuberculosis cases are much higher than other parts of the world. So you can see here that South Africa actually has the highest number of new cases per 100,000 population than any other country on earth. We have the African countries, of course, some of the Asian countries, some of the South Pacific. The Philippines, for instance, has a high prevalence and incidence of tuberculosis. India, which I've been spending quite a lot of time in and which we'll discuss, also has a fairly high incidence and prevalence as well, as do these other countries. So in terms of the worldwide incidence, you can see, again, the sub-Saharan Africa has a very high number of new TB cases, and this is pretty recent data in 2011. And some of the Southwest Asian countries do, or Southeast Asian countries do as well. And India, you know, has a high prevalence, but not the highest, unlike other countries, as you can see. And then, of course, North America and Europe, Western Europe, and Australia and New Zealand have very low new TB cases amongst their populations. So this is a picture of a TB research center in Chennai, India. And actually, I gave a lecture there a few years ago, and the lecture was actually on nutrition and HIV, but a lot of the investigators were also considering the co-infection of HIV and tuberculosis. And some of the research that I talked about in terms of using nutrition as a way to strengthen immunity was something that the other investigators in TB thought was important as well. And in any event, TB, as we saw earlier, is still an important disease in India, and by no means has it become eradicated. So tuberculosis in the United States, on the other hand, you can see is gradually going downward. And we had this critical time period in the late 80s and early 90s where we saw an increased incidence in TB. And really a large reason for that was because of the co-occurrence of HIV during that time. And as we said before, having HIV increases susceptibility to tuberculosis due to the impact on the immune system. So here is a graph of the percent of co-infection of individuals who are HIV positive in relationship to also having diagnosed TB. And you can see here again in the early 90s, the co-infection percentage was pretty high. It was all the way up to 30%. And this was among younger individuals. And then among all ages, it was, of course, lower. But still, it was quite a problem that concerned many investigators. But as we've developed medications, the ART medications to certainly provide to individuals who are HIV positive, because those drugs actually reduce the viral load and by doing that boost the immune system, then actually the co-occurrence here has gone way down because of it. So it really wasn't so much we were doing a great job in treating tuberculosis, we were actually doing a much better job of treating HIV. So what is the pathogenesis of tuberculosis, and why is it such a disease of concern even in today's modern world? Well, in terms of infectivity or infection, it's an airborne bacteria, and it's spread by airborne droplets. And generally, individuals who have an active form of TB are the ones spreading the infection, and it's spread by by individuals who have active TB, actually, coughing, clearing their lungs, and by doing that, they're actually liberating or releasing these bacilli through these airborne droplets. And in a susceptible individual, these bacilli are inhaled, they enter the lungs, and they go all the way to the alveoli. And the alveoli are at the very end of the bronchial tree, and that's where the oxygen exchange occurs. So what determines whether or not an individual will be exposed? What is their proximity to an exposed individual? How much bacilli was released? Again, it's the active infected individuals who are the ones who spread the disease. And is the recipient susceptible? Now, we learned with HIV that just because of the nature of that disease, they are very susceptible to infection. Now, once reaching the alveoli, these bacilli can multiply. And in some cases, in the active form, they can actually leave the alveoli and migrate through the blood supply to other tissue. Now, the alveoli, if you remember from your physiology course, or at least the anatomy and physiology lectures given here at educator.com, you'll remember that those alveoli, they're a single cell wall. They're like balloons at the end of the bronchial tree, and they're very highly vascularized with capillaries that engage in blood exchange. And these capillaries, of course, can also spread the disease. So that's really an important process or principle to remember in relationship to tuberculosis. So here's another picture of how indeed the tuberculosis is, of course, inhaled, and then it goes all the way down to the alveoli. Now, in most individuals have what's known as an inactive form of tuberculosis, and then a small percentage has what's called an active form of the infection. And that really depends on, does the tuberculous bacilli stay in the alveoli, or do they break out of the alveoli and migrate to other tissue? So this cavitation or cavitary TB is really an active form where we'll see that the macrophages and the bacilli actually create cavities where they can allow spread of tuberculosis through coughing. Now, fortunately, there's only a small percentage of active infection, although, because, of course, if there was a higher percentage, then more of the population would certainly be infected. So these bacilli that reach the alveoli begin, basically living in the alveoli, but also an important part of the infection is the involvement of these macrophages. So these macrophages, now remember, because the lung is exposed to the outside world, it's highly vascularized, but also there are a lot of lymph nodes in the lungs, and there's a lot of ways in which the lungs can certainly offset some of the pathogens and inhaled particles that can create injury and disease. So not only are the lungs highly vascularized, but they also have a lot of lymph nodes, and they have a lot of lymphatic interaction, so to speak, with the lung and the alveolus as well. So the tuberculous bacilli reach the alveolus, and then we see that we have resident macrophages, of course, and that these bacilli are ingested by macrophages. Now remember, the principle of the tuberculous bacilli are that they have these very resistant cell walls, and so they can be ingested, for instance, but not necessarily destroyed. In fact, some bacilli survive, and these bacilli are great. They do a great job of basically living. They basically live in the macrophages, okay? And because at this stage, the infection is truly confined to the alveolus, and there is no symptoms of disease that are present. Now, this is a more advanced stage, so to speak, of the disease, and you can see that more of these macrophages have come on the scene, and these tuberculous bacilli just love to live in macrophages, so they continue to multiply. So they multiply in these macrophages, and by doing that, they create a chemotaxis response. And from the immune system, you'll remember the immune lectures, you'll remember that these immune cells are very geared towards signaling from other immune cells. So these macrophages send out signals saying, there's an infection, come on board, let's get some more macrophages out here. And so additional macrophages migrate to the alveoli, and also other immune cells do as well. So at this stage, then we start seeing inflammation. So then we actually begin to see evidence of disease. And these macrophages actually can form a layer, and they can form what's called an early tubercle. And of course, they release cytokines and other inflammatory factors. And at this stage, you can also see the appearance of this sort of opaque area, I guess, so to speak, in the lungs if you were to do an X-ray. So at a later stage, after a few weeks, then we start seeing disease symptoms, and we have many macrophages dying at this point. And then when they die, they release bacilli, because remember, the bacilli are living in the macrophages. So the macrophages are not killing the bacilli. The bacilli are just waiting for the macrophages to die themselves, and then they continue to spread. And with this kind of death going on inside of this whole granuloma, is what you call it at this point, then you have this whole space developing, this caseous center, and then you actually have a situation where you begin to create an anaerobic environment. Now remember that the tuberculous bacilli is an aerobic bacteria. So, as happens in tumors also, you can have such a large growing tumor that parts of it, for instance, can not be exposed to oxygen anymore, and those parts can even die out. Well, the same can happen with this granuloma. So these aerobic bacteria do not do very well in the center, and so then they end up dying out, and these outlying bacilli and macrophages can then even form what's called a granuloma. And these granulomas can actually get quite large, and they can stay dormant for a long period of time. And also the other property is that they can actually reactivate at a later time. And at this point, then the disease is basically arrested. We have a latent granuloma that can stay around for a long period of time. So we saw that there was that little window of time where we had an active case of disease, but then it became dormant. So sometimes diagnoses of tuberculosis, for instance, can be missed, and they can become dormant and stay in that state for a very long period of time. Now, what about the active disease? Well, if you can imagine, if that granuloma, for whatever reason, becomes activated, it can rupture, and we can have what's called liquefaction. And that rupturing can then lead to dissemination of the bacteria throughout the lungs, into the circulatory system, and also into the lymphatics. And remember, the lungs are very highly vascularized, and they have an extensive lymphatic system as well. And this stage is known as active TB, and this is the stage in which individuals are infectious. So let's think about staging. Okay, we have stage 0 through 5. Now, this type of staging is not one like cancer, where as you go higher in stage, you indicate a more serious form of disease. Here, it's more like a timing of disease more than anything. So the real serious stage here is stage 3, where we have a TB infection and clinically active. Now, beyond that point, you can have a TB infection without being clinically active, and stage 5, you're suspected to have TB. So, of course, stage 0 is no evidence of disease. Stage 1 is being exposed, but no evidence of infection. Stage 2 is having TB infection, but no disease, and so forth. So with tuberculosis, you can go back and forth between dormant and active, basically almost throughout the lifetime of the individual, unless they are heavily treated with these antibiotic agents, and unless the disease is completely eradicated. So how do we determine that an individual has TB? Well, one of the ways, at least in the United States, is determining whether or not an individual has antibodies to TB by conducting this TB tuberculous skin test. And basically, the purified tuberculin protein is injected underneath the inner surface of the forearm skin here, and 48 to 72 hours later, that area is read to determine whether or not a skin reaction has occurred. And this is a picture here of what a positive skin reaction looks like. Now, if an individual, at least in the United States, is determined to have a positive skin test, they're often referred for an X-ray and for other lab PCR testing of the presence of the bacilli, for instance. So in terms of vaccination, at least in the United States right now, there's no vaccination. However, there certainly is a lot of research going on in terms of developing TB vaccines. In other parts of the world, however, the BCG vaccination is given, and it's given to young infants and young children on a pretty routine and regular basis. And the BCG vaccine protects not from infection, but actually from the active form of tuberculosis and another form of tuberculosis, a meningitis. There's a meningitis form of tuberculosis. So the BCG protects infants and young children from disseminated tuberculosis. And BCG would be certainly a solution, except that it doesn't last the lifetime. And BCG vaccinated individuals, if they travel to the United States, for instance, they will routinely test positive on the tuberculin skin test. So that's why, for instance, it's not encouraged for individuals in the United States to get a BCG vaccine, because then whether they're exposed or not cannot be determined, or whether they're active or not cannot be determined, because a positive skin test can mean either you've had a BCG vaccine or you actually have been exposed to tuberculosis. So the CDC has determined or decided many years ago not to recommend BCG vaccination for individuals in the United States, because the incidence and prevalence of TB, as we have seen, is very low. So, but the other important point is that BCG does not confer lifelong protection, that individuals who got vaccinated when they were young can still get the disease later on in adulthood. So I brought back this picture to show you that these are some of the agents that are being developed for treatment of tuberculosis. And really they're targeted towards different parts of the bacilli. So one couple of agents target the cell wall, another agent targets ATP synthesis. These other parts, targeting mechanisms that are not yet known. And then, of course, we can target the DNA transcription and translation. Now, we have a treatment regimen that's given for individuals with tuberculosis. And these are agents that are given for long periods of time. And in the United States, we also treat individuals who are demonstrated to have a latent infection even. So if, for instance, you're determined to have a latent infection, you will go through the treatment regimen of these antibiotic agents. So what we're seeing, though, in the United States is we're seeing these treatment-resistant, antibiotic-resistant TB. Fortunately, it's a very low level at this point, but we're seeing that this TB is not treatable with known agents, which is why we are certainly developing new and potentially more potent types of these antibiotics. So the first question that we're going to raise in today's lecture is, why is tuberculosis such a resilient bacteria? Number one is because, the cell wall. That cell wall does a great job of protecting tuberculosis, not only from the digestive enzymes of the macrophages and phagocytes in the immune system, it can also live in these alveoli for a very long period of time. It can also last for quite a while in the air as well. So the biggest protector of the tuberculin bacteria is really that cell wall. It's very, very challenging to break through with these antibiotic agents. So these antibiotic agents, they need to be pretty powerful. And the antibiotic treatment for the tuberculin bacteria sometimes lasts six to 9 months, and which is quite a long period of time. And these doses have to be taken every day. So, for instance, in the United States, this issue of compliance is really important. So at least in the health departments, in certainly local communities and counties, if they know somebody, for instance, is being treated for tuberculosis, they will visit that individual repeatedly to make sure that they are adherent with their antibiotics, because otherwise they can certainly become infectious, for instance. And if they don't take the whole regimen, what ends up happening? We contribute towards antibiotic resistance. So that these points are really important to remember in relationship to tuberculosis. So why did the incidence of TB increase in the late 1980s and early 1990s? Number one, because of HIV, that the HIV epidemic really came on the scene in the United States in the late 80s to early 90s. And at that time, we didn't have no treatment for HIV. And we will actually have another individualized lecture on HIV. So we're going to examine the disease from the perspective of HIV. But one of the things that we know about HIV, for instance, is it's a major immune suppressing type of disease. And because of that, other opportunistic infections that normally are kept at bay by a healthy immune system the individual becomes susceptible to these illnesses that normally they would have the strength to be able to overcome. So that concludes our lecture of tuberculosis for the day. Thank you so much for visiting educator.com.